Okay, it looks like most people have joined at this point. So hello, everyone. Welcome to today's IWGSC webinar series. My name is Joshua Stein, and I'm happy to be moderator today for today's webinar. I'm also project manager for the IWGSC. And I want to thank you again for joining us. Today we have our speaker, Sandip Kale, who will speak about novel sources of disease resistance in wheat. But before we start with the webinar, I'd like to just give a few introductory comments about IWGSC. Uh-oh. So, as you may know, the IWGSC is a nonprofit consortium with members from 76 countries, 3,450 general membership from including 943 institutes and companies and 8 sponsors. And I'd like to take this time to thank our sponsors that make this webinar possible. Arbor Biosciences, BASF, CIMMYT, Florimond Desprez, INRAE, Kansas Wheat, RAGT, and Syngenta. So thank you to our sponsors. And as you all know, the IWGSC was formed in order to bring the community a high-quality reference sequence for wheat, and we delivered that in 2018 with subsequent updates and currently version 2.1. Now the IWGSC is taking a turn to enhance this vision. So we are undertaking now a wheat diversity project with the vision to enhance breeding through an increased understanding of the molecular basis of traits and their allelic diversity. And starting this January, we initiated a 2-year funded project that is a collaboration with funding from the US and European Union to assemble platinum assemblies for 12 bred wheat landrace accessions and a renewed Chinese spring sequence. And with that, we will also be producing transcriptome data, gene annotations, and an update of the Chinese-seq RefSeq, both the RefSeq and annotation. And finally, these data will be used to build a practical haplotype graph, which is a tool that will enable us to catalog all the genetic diversity within bred wheat. And this is also a tool that's very useful for imputation of genotyping to apply to various breeding applications. So please stay tuned. This project will be releasing data over the next year and a half. Okay, I'd like to update the community with regard to IWGSC events. So there will be an IWGSC workshop at the Plant and Animal Genome Conference in September in Perth, Australia. We have a great panel of speakers lined up. They're almost nearly finalized. I would definitely look forward. I will be there. I would look forward to seeing all of you there as well. So I hope you can make it. As far as future webinars, our next scheduled webinar is on October 17th. So I hope you can join us for that. We'll be joined by presenter Yves de Smet of the VIB UGent Center for Plant Systems Biology. So please look out for that email and register if you can and attend this webinar. Just a quick intro to how this webinar, the interface for the webinar. There is a chat, but that chat is used to communicate messages from the organizers of this webinar to the audience, and that might include handouts or other information that you might need. If you have a question that you would like to ask, please use the Q&A icon and submit your question, and those questions will be answered with any remaining time at the end of the webinar. And as well, Sandip has agreed to any questions that he didn't get to, he will try to answer in written form. Okay, so let's move forward then. I'm very happy to introduce Sandip Kale. He is Assistant Professor of Crop Genetics and Biotechnology in the Department of Agroecology at Aarhus University in Denmark. And the title of his talk is Enhancing Crop Resilience, Genomic Insights and Novel Sources of Disease Resistance in Winter Wheat. So I thank you again for your participation and being a great audience. Thank you.
Yeah. Yeah. Thanks, Josh, for the introduction. And yeah, good morning, good afternoon, good evening to all, based on the work zone you are based in. It's my pleasure to present the work I did when during my postdoctoral tenure at Leibniz Institute of Plant Genetic Research, IPK, Germany. So before I start my presentation, I would like to give a big background about myself. So yeah, as you know, my name is Sandip Kale. I did my MSc and PhD in biotechnology from India, and then I was working as a project assistant with International Crop Research Center in ICRISAT in India. And during my PhD, I also worked for 1 year as a visiting scientist in US. And then I moved to Germany to work on wheat. It's a gene bank 2 project where we characterize all the winter wheat accessions from the IPK gene bank. Recently, I also worked in Carlsberg for 2 years, and now I'm working as assistant professor in crop genetic and biotechnology section at Aarhus University. Yeah, I'm really fortunate to get an opportunity to work on different crop species, starting from linseed, which is an oilseed crop rich in omega-3 fatty acid, and then at ICRISAT I was working on chickpea, which is a legume crop, and recently since my postdoctoral tenure at IPK, I'm working on cereal and mainly on wheat. So today's work today I will present our work which is published. So this is the first paper where we describe the genotyping of 9,000 winter wheat taxation and its used for identification of introcreations. And then in the second article, we talk about the development of core collection and sequencing of a resistant gene using resistant gene enrichment sequencing, and its use for identification of candidate gene for yellow rust and leaf rust in winter wheat. And then I would like to also thanks all the team from this Gene Bank 2 project, Dr. Johan Rep, who was the principal investigator, along with Neil Stein and Martin Marsher, and I and Albert. We mostly perform all the analysis in this project. Albert was looking for the phenotyping analysis, while the genotypic analysis was performed by me. And then I also like to thank Dr. Brande Wolf for his assistance in analyzing the resistant gene enrichment sequencing data. So I have outlined my presentation in 4 topics. First, I will describe genotyping by sequencing of 9,000 winter wheat accession and its use for studying the diversity. Second, I will talk about phenotyping of this 9,000 accession and construction of trait customized core collection. Then I will talk about the whole genome sequencing and its use for identification of introcreations, and finally about resistant gene enrichment sequencing and candidate gene discovery for yellow rust and leaf rust resistance in winter wheat. I think there is no need to give more introduction, but just to start with I got this slide from picture from the International Wheat Genome Sequencing Consortium, which shows the evolution of the wheat. As you know, wheat is the hexaploid crop, which was derived from this diploid progenitor by 2 rounds of polyploidations. It has 3 subgenomes, A, B, and D. The genome size is 15 GB, and total it has 21 pairs of chromosomes, 7 from each subgenome. Wheat is a really important global crop, providing 20% of calories and protein requirements. But also the wheat yield is severely affected by different wheat diseases, and mostly caused by fungal diseases. And so there is a need to identify genes and which can act against these diseases and improve the yield. So and then first thing for identifying this novel source of candidate gene is the plant genetic resources stored in the gene banks. So during my postdoctoral tenure, I was working at IPK, which hold European Union's largest gene bank, which has around 28,000 accessions. In the Gene Bank 2 project, we aim to characterize 9,000 winter wheat accessions. This slide shows the distribution of 9,000 winter wheat accessions all over the world. As you can see, mostly they were from the European region, but you also have good representation from other countries like South Asia and also from North America. You can also see there is a large amount of diversity in the spike traits, but it was also showing there was also diversity in the disease response. So yeah, in order to activate characterize this 9,000 accessions, we used a 2-step strategy. So the seeds from the gene bank were taken out in the batches, and then they undergo 2 rounds of single cell descent before going for DNA extractions. So this single cell descent was introduced to achieve the homozygosity and remove the mixing or any heterozygosity present in the seeds. And then we use the 2-enzyme restriction digestion protocol for developing the genotyping by sequencing library using the PST1 and MSC1 enzymes. And then around 540 digested and ligated libraries were pooled, and approximately 2.50 million reads were generated per sample. And then we use open level available softwares for identifying the SNPs. For this, we first remove the adapters and low quality bases from the short reads, and then the reads were mapped against the reference sequence of Chinese Spring and used for SNP identification. As I mentioned, the reads were mapped against the reference sequence of Chinese Spring, and first thing what we did is to see the mapping quality, and as you can see, with 99.9% accuracies, more than 60% reads were mapped on the genome. But when you look at the each subgenome-wise mapping, around 20% reads were mapped on each subgenome, but there were few accessions where very low number of reads were mapped to the D genome. And then when we looked into the details, we found that there were hardly read mapped on the D genome. So these were the potential tetraploid accessions which were misclassified as the hexaploid. So so this was the first achievement with this data, that we could correct the errors in the passport. Secondly, we use these SNPs to study the neutral diversity present in the 9,000 accessions. So we first conducted the principal component analysis, and as you can see, first principal component separate the accessions from South Asia and the Europe, while the second principal component separate accessions from East Europe and West Europe. We also found that this accession from North America, they were clustered close to the East European accessions, which also relate with their history of breeding and involvement of European accessions in the American breeding program. Then we also perform a population structure analysis and identify 5 subpopulations, each corresponding to these subregions, which we identify from the principal component analysis. And then we use this similarity within the populations to identify the diversity present within each subpopulation. The x-axis shows the similarity while between the each pair, while the y-axis is the just density. And the broader the base means the higher is the diversity. And you can see the accessions from population 2, which correspond to Southern Europe, and population 5, which is from the South Asia. They were highly diverse compared to rest population, which also indicate use of those accessions in improving in the European plant breeding program. We also identified some accessions which were highly identical, and then we did this similarity-based analysis to identify the duplicates. And from this analysis, we found around approximately 20% duplicates in the IPK core collection, At the same time, there was around 4,000 accessions were from INDRA-E, were also sequenced using the SNP array. And then we developed pipeline to compare the diversity between these two core collections, these two corrections. What you can see from here, this plot, as both the collections has almost same diversity, but there were also some gaps. For instance, here this region is missing in the IPK collection, while this part which was lacking in the intra collections, which indicates that there is need to fulfill these gaps so that we have backup of accessions in both the collections. And now, as I mentioned, wheat is though important, it's severely wheat yield is affected by different diseases. And in this project, we targeted three diseases: leaf rust, which is also called as brown rust, powdery mildew, stripe rust, and the stripe rust. So in order to phenotype this collection, we use two-stage strategy. We took out 1,600 plant samples in one batch. They were grown in one row, and then they were phenotyped there. And then the single spike was harvested from this year, which was grown in next year, and then again phenotyping was carried out at this second year. In this way, we phenotyped 6,000 accessions, and the heritability for this I'm showing for yellow rust, but it was also good for other two diseases. And then the next obvious step, once we have this genotypic and phenotypic data, is to perform genome-wide association analysis. And as you all know, that this standard reference-based pipeline, where you map the reads against the reference genome and identify SNPs, and those can be used for performing the genome-wide association scan. But it has some limitations. First is, as we all know, that single assembly is not enough to capture all the variation present within the species. There can be also errors in the assembly, and that it might be because of technical or during the assembly process. And also we also know that from these multiple genome assemblies that within species there are large structural variations which cannot be captured using the short read data. And that's why recently many groups are developing reference-free approach, wherein raw reads can be directly used for identifying k-mers, which are nothing but short stretch of unique sequences. And then these k-mers can be used as a genotypic matrix for performing GWAS. And once we do the GWAS, significant k-mers can be identified. Those can be mapped to the multiple assemblies, and also or can be assembled de novo to identify the candidate gene. So we use this k-mer-based GWAS approach, which was published by or was developed by Dr. Professor Wiegel's group, which uses raw reads, which were high quality raw reads for identifying the k-mers. And then based on this k-mer counts, a presence absence matrix is developed, which can be then combined with phenotypic data. the main challenge with this k-mer based approach is you get billions of k-mers, and then it's highly computationally challenging to perform genome association for each k-mer, and that's why they have this permutation based strategy to identify top 1 million k-mers, which can be used in this linear mix model for doing the actual genome association studies. Here I'm showing this result for yellow rust, where we use we identify k-mers from 4,635 accessions, and after performing GWAS, 21,000 significant k-mers were identified. When we map these k-mers against the Chinese Spring reference assembly, we found already known region on this 2A, which is the wire 17. But there was no other positive hit. But when the same 21,000 k-mers were mapped against the reference assembly of SY Mattis, we found the same peak on this wire 17, but also found a strong peak on 6A. This indicates that this 6A peak, this region is missing in Chinese Spring, and that's the advantage of this reference-free approach. I will talk about this 6A when I'm presenting the result from the resistant gene enrichment sequencing. Because we did this phenotyping for all these 6,000 accessions, but it was not possible to do this multi-year, multi-location phenotyping, and that's why we decided to construct a trait customized core collection. We use this against two-step strategy. In the first step, we use the phenotyping data and identify top 200 susceptible and resistant accessions, and then the phenotypic data was combined with the genotypic data, and then 150 resistant and 50 susceptible accessions were identified for each diseases based which were covering the diversity present in the entire collection. And then in this way we generated a core collection of 800 accessions, which consist of 200 European elite lines and then 600 plant genetic resources. We included these European elite lines in order to have these known sources of resistance. And then for the 600, there were 150 resistant accessions for each diseases, while 50 susceptible accessions. And then this data, we phenotype this core collection for 7 different environment, and you can see there was clear enrichment of resistance allele in this the accession from trait customized core collection. We sequenced this core collection at higher depth using whole genome resequencing approach, and then it was used for further identifying the intrications. So as I mentioned, breed wheat is derived from its progenitor by 2 rounds of polyploidation. But the beauty of wheat is it can still be crossed with its progenitor. And there are 300 different species with wheat, winter wheat can be crossed. And these are called as alien intrications. And breeders has used this wild relatives of wheat for interrogating the disease resistance or important agronomic reports. However, these interrogations are not documented properly. So it is needed. That's why we develop pipelines to identify those interrogations. One of the known interrogation is 1B interrogation of 1R Y chromosome in the 1B. And then there are, based on this reference assembly, we also identify, based on the reference assembly of 10 wheat genomes, they also identify more interrogations on different chromosomes. And then we decided the simple workflow just based on the sequencing data to identify the interrogations, because these interrogations, these regions are diverged from the bred wheat, So when you map the reads from the introcations on the Chinese spring, there is a drop in coverage, and we use this drop in coverage data to identify the introcations. This is one of the example for the one of the winter wheat variety. You can see the normalized coverage all over the chromosomes, but you also see drop in coverage on different parts of the chromosomes. For instance, here, these 2A introcations, you see drop in coverage, and we use this the coverage data to identify the introcations. And by this analysis, we identify introcations, 8 introcations, and we studied the frequency of those introcations. As expected, those introcations were present in high frequency in elite lines compared to the plant genetic resources. We also classify those this all the accessions based on the year of release data, and again did the detailed analysis of those introcations. And you can see the introcations were they were present after the green revolutions, but there were very few introcations or no introcations before the green revolutions, and they were also positively selected, and then that's why they are present in very high frequency in the current elite lines. One of the introcations we identify was on this pore. It was the introgation on long arm of the pore, and another study found that it's integration from triticum dicoccum. We studied the frequency. Again, it was positively selected in the elite lines, and our group also carried out the genomic association study using the elite lines, the hybrids of the elite lines, and identify a strong peak on the chromosome 4A. But that time the integration was not known, and then we thought that it's related to this LR34B. But when after the identification of this integration, we can clearly see that the resistance in this elite line is because of this integration and not because of this LR34 gene. Yeah, so that was about the use of whole genome sequencing to identify the integrations. And then our last part is use of this resistant gene enrichment sequencing to capture the NBS-LRR genes. As you know that most of the cloned genes are of NBS-LRR categories. And if you see this, the earlier work, these resistant genes, they show present substance variations. And from our data also, we found that the single assembly is not enough, and also it's not possible to develop hundreds of assemblies, hundreds of chromosome scale assemblies. And that's why it's this resistant gene enrichment sequencing, which aims to capture only the NBS-LRR part, is efficient for identifying those resistant genes. So we did this resistant gene enrichment sequencing of all these 800 accessions from the trait customized core collection. It has 2 steps. First is the construction of library, preparation of libraries, and then in the second step, baits are used to capture the NBS-LRR containing fragments. We got this bait from Dr. Brande Wolf. And then we use this. Once we get this, we sequence this range NBS-LRR fragments using 2x250 parent data, and then the data was analyzed to identify NBS-LRR using de novo assembly pipelines. And then we use this, we study the different domains present in this NBS-LRR. As expected, we found mostly coiled coil NBS-LRRs in our data set, which is the characteristic of monocots. And based on the pan-genome analysis, we know that there is in each species there is a region which is shared in all the accessions of the species. That's called core region. But each species also has individual, unique, or dispensable regions. Few years back, the attempt was carried out to identify or generate this pan-LRM using the same pan-genome concept to identify core NBS-LRR and this cloud NBS-LRR, which are specific to each accessions. We also try to attempt to develop this parallelogram in wheat. So there were total 1,053,1.5 million invasive larvae were identified from these 800 accessions, and then they were clustered into 10,000 different clusters. Unfortunately, we could not identify the core region. We could identify the cloud, but we could not identify the core. And this may be because of the use of this short read sequencing, which was not able to capture all the invasive LRR, or maybe this highly related invasive LRR may get collapsed during the assembly process. But we got the data to predict how many accessions are needed to capture the entire NBSLR from this winter wheat. And we use the cluster, what we identify to perform the saturation analysis. You can see on this x-axis, these are the number of accessions, while y-axis shows the clusters discovered. And when we consider all these 800 accessions, the 95% NBSLR can be captured using around 65 random samples. When we consider only plant genetic resources, we need around 80 random accessions to reach to this 95% saturation level. While with elite lines, we need around 135 accessions to reach this 95% saturation, which indicates that elite lines has low invasive LRR diversity compared to the plant genetic resources. But another important point is these three curves, they never converge, which means that there is something unique in the elite lines which is not present in the plant genetic resources. To identify that reason, we identify a number of resistant associated cluster from each accessions and then compared against the disease susceptibility. Here I'm showing for this yellow rust, you can see that elite lines, they have more resistant associated clusters, and then they were also low susceptible to this yellow rust. When we see the distribution of these resistant associated clusters on this 3 subgenome, we observed that those resistant associated clusters in elite lines, they were coinciding with the introgase region. And from this analysis, we conclude that elite lines has this introgation region, which were selected for this disease resistance, and that's lacking in this plant genetic resources. So in order to construct the pan NLRM, we need both this plant genetic resources and also the elite lines. Based on our earlier analysis, we found that attraction has this introgation on this chromosome 4A, but it also carries this 6A locus for yellow rust resistance. And that's why we decided to develop a chromosome scale assembly of attraction. We use this PacBio high-pi sequencing plus Hi-C data to develop the chromosome scale assembly. The quantity gain 50 was really high, around 18 MB, which was higher than all these 10 plus assemblies developed using the Illumina short read data. And then using the Busco analysis, we found that 98% of full length genes can be captured in this attraction assembly. We also performed the synteny analysis with the Chinese spring assembly, and we found overall good correlationality between the two assemblies. But we also identify some intrications, for instance, here the inversion on 4B, but also on this 4A. Then we use this phenotyping data for yellow rust and leaf rust to perform genome association analysis. We performed GWAS using both reference-based and the reference-free approach. Here I'm showing the reference-based approach. In the first plot, the SNPs were identified by mapping reads against the Chinese spring, while in the bottom part, the SNPs were identified by mapping read against attraction. And and this is the result for the yellow rust. You can see the strong peak on this 6A, but there were also peak on other chromosomes. When we perform the reference-free approach, we identify the significant KMers, and then they were mapped to both Chinese spring and also the attraction. And we identify this peak on the 6A. But in Chinese spring, you see mapping on all this homologous chromosome 6A, 6B, and 6D, which also indicate that this region was absent in Chinese spring. To test it, we did the coordinate analysis between all these 10 plus assemblies, and we found that this region was only present in SY methyls but was absent in all other assemblies. And again, we checked the frequency of this region, and it was highly selected in the elite material. Same analysis, we did it for a leaf rust. Here I'm showing the data mapped against the attraction. And from this analysis, we identify a strong peak on 4A, which was related to the introcations. But we also identify other peak on this 5B and also on the 3D. Yeah, as I mentioned, the peak on 4A was related to the introcation. And we performed this microsynthetic analysis for the region on this 5B. And we found that this region was only present in the attraction and was absent in another cultivar assemblies. We also carried out phylogenetic analysis with known resistant genes already known in the wheat, and the NBS LR from this 5B, they were closely related to this LR21 gene reported earlier. To summarize my talk, I hope I convinced you that the G-based analysis was helpful to activate the European Union's largest winter wheat collections. We also developed pipelines to compare the diversity between the gene banks, also identify the introcations, and then study the involvement in wheat breeding. And from the rain-seq analysis, we could, although we could not generate the panel around, but we now have the estimate of number of accessions needed to capture the entire NLRM from winter wheat. And from this reference-free approach, we identify novel region for yellow rust and leaf rust resistance in wheat. Now, this is the last slide, but I'm also using this correct core collection for studying the nutritional quality, where I'm currently working on identifying the region for grain protein content. And so there are recently there is a opportunity for doing Marie Curie postdoctoral fellowship. Also, we have there is opening for Novo Nordisk Foundation's postdoctoral fellowships. So if you are interested, you please contact me. Thank you all for listening, and I'm happy to answer if you have any questions.
Okay. Thank you so much, Sandip. That was a great talk. One comment from BD says, excellent work, Sandip. So there's agreement in the audience. I will start with, there are a couple of questions from Vijay concerning how you do the phenotyping and scoring for leaf rust.
Yeah, so that was we did it the field phenotyping using the standard scoring methods.
Okay. And so standard scoring method, that was the second question. How many replications and environments were considered for phenotyping in GWAS?
Yeah, so in the beginning, when we were doing this, when we phenotyped all the 6,000 accessions, that time it was only phenotyped at one location, but for 2 years. But the score collection, it was phenotyped for multiple for the 7 different environments.
Okay. Next question, which gene is corresponding to the YR locus that you identified?
Oh, that is 1 MB region actually, and it has around 9 NBSLR genes. And we could not identify a single gene, and I'm currently working on further characterizing and identifying the candidate gene for yellow rust.
Okay, next question. The SNP identified in winter wheat may also be associated with rust resistance in spring wheat. Do you have any thoughts
Yeah,
on that?
yes, the 6A locus. Yeah, recently there was a study published from a group in Norway, and they also identified this locus, 6A locus in spring wheat population.
Okay. Next question. In your opinion, which model, Gapit or Tassel, is ideal for GWAS?
Yeah, basically both are developed by the same group. So, and then there are basically there are different model available, uh, and based on I think this both softwares are doing good. But when you are doing this reference P analysis, there is a limitation of this computational resources, and that's why you need to use different softwares. In our case, we use the Gamma.
Okay, thank you. All right, next question. Hello, and thank you for this interesting exposition. Two brief questions. You made the PCA gene bank and the other PCAs in R, or did you use a different program?
Yeah. So all these analysis, yeah, most of the analysis was performed in R.
Okay. And then what is your opinion about the UAV drone image technology for phenotyping?
We haven't tested it, but yeah, I think that's the future. We need to use this drone imaging for, yeah. You cannot do it for the all, but for this trade customized core collection, that can be definitely used.
Okay. That's all the submitted questions so far. I have one additional question. The introgressions in the modern wheat lines is very interesting. I think you mentioned you identified the source of the introgression from one
Yeah.
of those loci, and I'm curious if the other loci also came from the same source, so that the introgression, occurred, via one breeding program that introduced these introgressions, or you're finding these are multiple events from different sources?
Uh, yeah. So these integrations were from different sources. For instance, this 4A integration, which I mentioned, that was from dicocoid, but that 2A, which is a well-known integration from Timo Pivi. Yeah, I actually don't know the exact name of all the integrations, but yeah, it's all mentioned in our article.
Okay. Okay, here's a question. Will it be possible to use this approach in order to detect genes underlying QTLs?
You mean this reference pre approach? Yeah, definitely. Yeah, because this is just the new approach, but you can basically use for any trait.
Okay. And we have an additional question. Can you repeat the name of the software that you used? Is it free?
Yes. We use all these open source softwares. They are all free. So for the GWAS analysis, I use this Gamma.
Okay, thank you. Okay, if there are no other questions, okay, I believe all the questions have been answered. So once again, thank you for a great talk, a very informative talk, and interesting talk. And I'd like to thank you again, all of the attendees, and we will see you next time for our next webinar in October. So look out for that, and thank you again, Sandip, Kale. Appreciate it.
Yeah. Thanks for providing me opportunity.
All right, terrific. Thank you.
Yeah, bye. Shall we